So this is Neurophysiology Notes number 1, and the first thing that was covered were the three major debates. So the first thing was the mind versus brain debate. Are the mind and brain separate or unified entities? So back then, neuroscientists were really having this philosophical issue of whether the mind comes from the brain. It seems easy now, but back then they truly didn't know. And then cases such as Phineas Gage showed that indeed the mind does come from the brain, because if you damage the brain, suddenly the person becomes affected. They almost become a different person. So the two are indeed connected. So the next one is localism versus holism. Do neurons and brain areas have specific functions, localism, or is it undifferentiated working as an aggregate field, holism? So I guess you could say it's kind of both, right? Because in the real world, from what we know, our brain has specific areas where it uses, it has a localized function, like Broca's area or the occipital lobe for vision. However, the entire brain does work as a unit, helping each other. So it's not necessarily independent localism, but it's not necessarily completely together, meaning that every part of the brain works together for one goal. It's kind of interdependent, where you have various parts of the brain functioning and having one unique task, and then combining the efforts as a whole, as the brain. So the next one is the nature of neural communication. So does the brain create the mind, and can consciousness be simplified to neurons? I guess this is a philosophical question, right? Because can the human brain, can we just justify that neurons are the ones that create consciousness? And I think we can safely say that, because if we destroyed a brain, for example, in the famous case of Phineas Gage, the person, that their consciousness becomes different, their personality changes. What makes them changes, and so we can say that the brain does create the mind. So moving on, energy is needed to separate opposite charges across a membrane, and if opposite charges are separated, the system will have potential energy. So this is just talking about the membranes of cells, and how, for example, in a neuron, we have the Na potassium ATPase pump that pumps out sodium and pulls in potassium. And so this helps create that difference of charge, and so charges are separated. And so it creates this potential energy. So you have a larger concentration of sodium outside than inside, and so it creates this potential where the sodium can rush in, just like you see in action potential. You can think about it as if, for example, a waterfall. You have the potential energy of the water at the very top, about to fall down, just like how in a neuron you have so many sodiums outside ready to come inside if the gates open. And that leads us to our next topic, which is the ion channels have basic functional properties. And these ion channels are what allow the ions to come in or out, depending on the ion. And so they basically have three main properties. Their properties are to gate, to select, and to conduct. So gate, obviously, you have to create that potential, and so for that to happen, the ion channel has to be closed, and so that's what it's doing at rest. For example, for a sodium channel, voltage-gated sodium channel, and so it it has to be specific in the fact that it has to only let sodium in, because if it let other ions in, it would mess with the entire system. And then finally, to conduct, you have to be able to have these channels to conduct these ions, to allow them to flow inward, and to allow them to cause the difference in potential to change, and therefore help create the action potential. So moving on, we have two main types of ion channels. We have leakage and gated. So for leakage, we have, for example, potassium channels. Potassium channels are always open, and so potassium ions are free to move in and out as they want. Gated ones are like the voltage sodium channels that I was talking about, where you have part of the channel, the protein, that will open or close depending on different aspects. So, for example, this illustration shows that in an open a place, the proteins will be apart so the ions can flow, and then in a closed, you'll have part of the proteins come together to help make sure that ions can't come in or out. And so, and then the different types, right? So this one, you have part of the protein come in or come together to help close the membrane to prohibit ions from passing. And in another case, like the sodium channels, you have a little ball and chain that will help swing up and then prevent ion movement. So the 5 types of gating controls. So the first one that we'll talk about is ligand binding. So you can have a closed conformation, and then a ligand will come in, fit into the gap, right? So it's a triangle and triangle. So it's like a key and lock mechanism, and so it will fit in, and then it will change to a open conformation, and then it'll allow ion passage. The next type is phosphorylation. So phosphorylation can come along and then attach to the ion channel and then cause it to change conformation into a open state. Voltage can do the same thing. You can have a voltage sensor that can sense the voltage changing and therefore change its conformation in response to an open position. You can have mechanical force gating, so stretch receptors, for example, in your skin, they can stretch, right? You can stretch your skin, and that will cause tension on the cytoskeleton to pull on the ion channel, so it will allow ions to conduct and therefore cause an action potential, and then therefore lead to the mechanical stimulation causing a action potential and transferring that into sensory stimuli. And so the final type is temperature gated. So you have temperature that's causing it to change from a closed to a open position. Now I found temperature pretty interesting, is if you think about it, what exactly is temperature, right? It's just, for example, it's an increase in movement of the air molecules, but how could that change an ion channel? So I was puzzled about it, so I Googled it. So apparently, well, just some background info, an example of temperature gated channels are trip channels. For example, trip V1, which detects noxious heat. It's also the one that is activated by capsaicin, the compound found in chili peppers. So what happens is it's similar to how as temperature increases, an ice cube melts. So the actual thing changing is the protein itself in the membrane. So the protein, as the temperature increases, the protein itself changes in response to the heat. For example, just the same way at a certain temperature, the ice cube starts to melt into water. Same thing happens with this protein, and that helps it transfer that potential. It allows that conduction or transduction actually into an electrical signal, therefore allowing our bodies to detect that heat. So I thought that was pretty interesting. But yeah, these are the 5 ways that ion channels can be conducted, or the types of gating controls. So I hope that helped, and thanks for watching.